Okay, so welcome back to 返學 DSE, and we are now in chapter 25 talking about genetics. So first of all, let me talk about how genetics and biology works for me, because it is a very fundamental concept, but we only taught in a very late stage of our studies. So to me, biology is a study of lives, and how can we study lives? We study by comparison, that is, studying how we are different from each other. And actually, genetics is a study of inheritance and also variations, which is a fundamental tool in understanding biologies. We are finding things that are similar but not identical, and we look for the difference between them. And here is a classic example of the mutant ninja tortoise that is owned in Chinese called 忍者龜. Okay, so it is an example. So they are different from the normal frog. So what is the no frog is a turtle, sorry, yeah. So they're different from normal tortoise. So how they're different? What makes them so different? Is that because of something that inherited or something causing them to have a mutation or what? So we would like to understand that. And also there is a very historical example about the Drosophila, that is the flies. Okay, normally for flies they all have red eyes. Okay, all of them are red eyes. However, some days someone investigate and then they found out there is a fly that is in white in color with their eyes. So they ask a question. So what happened to that particular fly? So based on that white-eyed fly, we have known that for the Drosophila, the wild type, that's the normal one, is the red eyes, while the mutant one have the white eye. And it also starts the study of the genetics. In order to study genetics in our curriculum, we have to understand about Mendel's and his laws. So for the Mendel's law of inheritance, there are two contrasting traits in the parents, and only one appearance is shown in the F1 generation. This means that for the parents, they cross, and in the F1, there's their first offspring, only one appearance is shown. And then for this F1, they cross with each other. And then the missing traits reappear in F2 in a ratio of 3 to 1. So in short, Mendel's first law is what we call as the 3 to 1 ratio. So how can this happen? So in order to understand it, we need to use some terms. So terminology is very important for genetics. So you have to understand what terms you are using. So for genes is the basic unit of inheritance, and it controls particular traits. And because we are all diploids, we have a homologous chromosome, so which means that one gene will have two copies, and each copy may be the same or different, and they are called as alleles. So we have two copies, allele 1 and allele 2, and they're in the same gene. And on a DNA, we have a lot of genes, and in a chromosome, there are so many DNAs. So let's focus on a gene called gene T. Okay, if both alleles are the same, which known as the 大 T 大 T and 細 T 細 T, we call them the homozygous. And if they are different, which means there's one big T and one small t, 大 T 細 T, we call them as heterozygous. And we don't call it as like TA or TB. We only call it as 大 A 大 A 大 A 細 A 大 A 大 B. Never happens. Why? It is because for the same gene, they have two alleles. In this case, big T and also small letter t, and they should be in the same letters. So that's why we don't have 大 A 大 B. We only have 大 A or 大 A 細 A. So let's return to the Mendel's law, that is the 3 to 1 ratio. So let's say there is a gene called gene T controlling the height, and we have three different combinations. We know as the genotype, that is the 大 T 大 T, 大 T 細 T, and also 細 T 細 T. And we have two phenotypes, that is something that you can observe. For the 大 T 大 T and 大 T 細 T, they all appear to be tall, which we term them as dominant phenotype. Okay, and we also term the allele T, big T, is dominant. Okay, while if you have small t, we call it as short, which is a recessive trait. Okay, and then if you want to know more, you may go for this video in TED Ed, and you will know more about how his experiment is. So in order to start this particular chapter, you have to know how to draw a genetic diagram, and in order to draw it, you have to term the terms first. That is, what is 大 T and what is 細 T. In here, 大 T means the allele corresponding to the tall plant, while for the small t is the alleles for the short plant. And for the parents, they are there's the F1. They have the T 大 T 細 T and 大 T 細 T, and their gametes will be shown in here. And then their offspring will shown in this combination. And as long as you have one big T, no matter you have one or two, as long as you have one, okay, you will appear to be tall, which is a dominant trait, okay? While if you have both of them are in small letter d, the side T, you appear to be short, which is a recessive trait. Then amazingly, this is 3:1 ratio, which is the same as the observation. And remember, for the outcome of any course, it is not related to the another one, so we know as the independent event. And you know this concept in your core mathematics in probabilities. In a simple example, that is a floating a die. Okay, if first time you float is at 6, for the second trial, what is the chance of having another 6? It is still 1/6 because the first trial and the second trial are independent event, which means that they are not interrelated to each other. So for the first time you try at 6, you may think the probability is 100% getting 6, okay? However, if you have an enough sample size, let's say 600, okay? And then the chance of having a 6, that's the times of having a 6, should be around 100. This is a concept you learn in core mathematics about probabilities and your expected outcome. And we have to identify the genotype, because for the phenotype, this is something you can observe, while for the genotype, you have to identify by crossing. So how can we do it? We do it by test cross. Test cross is done by crossing with a homozygous recessive individual. Remember, for homozygous, it only means the two traits are the same, either 大 T or 細 T. But if homozygous recessive, there is only one possible, that is 細 T. So let's say you have a tall plant, so you may either be 大 T or 大 T 細 t, and then you cross with small t. So if all the F1 are tall, then your genotype should be 大 T. If 50% are tall or 50% are short, then you should be 大 T 細 t. That is what we call the test cross with the homozygous recessive individual, and you should know how to draw it in a genetic diagram. Okay, try it now, please. Okay. If you're not sure, then you may go for your book, because in your book, it already drawn once how it can be done. So for the Mendel first law, it is controlled by one gene, two alleles rule. In short terms, there is 大 T, 細 t, and 細 t. And there is another rules called Mendel's second law. So there are two genes on different chromosomes, and they have contrasting traits, and each of them will have two alleles only. So there is an example, 大 T, 大 Y, 大 T, 大 Y, 細 Y, 細 T, 細 Y, 細 Y. Okay, you may try to draw it, and then you can have an outcome. So because of the separation of the allele is an independent event of each other, that means that the gene T and gene Y are independent, and then you will have a ratio of 9:3:1, in Chinese called 9:3:1 rules. Okay. So you have 9 for anyone with a big T and then with at least one big Y. 3 of them have a big T and then with 2 Y. 3 of them will have small t, and then with at least one big Y, and then 1 will have all in small letters. Okay. What is this used for? It is used in a game called Animal Crossing. Okay, a famous Nintendo game. Okay. So how it can be done? So here an example how Animal Crossing can be done. So in this game, you are going to grow some flowers, and all of the players are interested in growing a blue rose. So how can you get a blue rose? You have to understand the genetics behind. So there is a gene called redness. So if you have a big R, then you are red. If you have 2 small R, and then you are not red. And you have a yellow, okay, there's a yellow gene called Y, and you have as long as one Y, and then you are yellow. If you have both small letter Y, and then you are no yellow, okay. And there is a white gene. If you have one big letter W, and then you have white, and you have both a small letter W, and then you are not white. Remember, W is not a good letter in doing genetics, because it is sometimes very difficult to tell a big letter and a small letter one. And similarly for the S. But for the S one, it includes a more advanced concept called the epistatics, and it is not required in your curriculum. However, if you want to get a blue rose, you have to understand the genetics behind. And I once tried it, and I worked it out for my wife, and it spent me 3 hours. And this is okay. It is complicated, okay, and I enjoy the process because it's not a lot of people in this world can do it, okay. So back to your curriculum. So for the inheritance in human, we have to understand 3 particular examples. One is the ABO blood group, it involving 3 alleles, IA, IB, and also small letter I. Sex determination, which means that the females are XX while the males are XY. And based on that, we move to the sex-linked traits because they do not follow the Mendel's law. And then your mother will determine everything, and usually the boys will suffer. This is the ending conclusion. Okay. So let's start with ABO blood group first. There are 3 alleles for the antigens on your red blood cell, IA, IB, and small letter I. And then it will result in different blood group. Let's say for blood group A, you may either the genotypes be IA or IA small letter I. And with at least one IA, and then there will be antibodies present in your plasma. That is the anti-B because you don't have antigen B, so you have anti-B because you don't want to kill yourself. Okay, if you have small letter I only, and then both anti-A and anti-B will be found in your plasma. So for blood group O, they have both anti-A and anti-B, and blood group O is known as the universal donor. If you want to know more, you may also go for a famous clips about Stephen Chow. That is the 滴血認親. Okay, it's very famous. If you just use this term and go for the YouTube, and then you can find the related videos about 滴血認親. And for the sex determination, for human, we have 23 pairs of homologous chromosomes, and for 22 of them are called the octosomes, 常染色體, which means they are not related to sex, and there are only 1 pair of chromosome corresponding to our sex, and we call them sex chromosome. For males, it's known as the XY, and then for the female, they are known as XX. So for the X chromosome is longer, and it will also carry more gene. And then for female, they have 2X, and then for male, they only have 1X, and that particular X must be inherited from the mother because the Y is inherited from the father. So let's have a look in the sex-linked traits. In this example is the red-green color blindness. Usually for the sex-linked traits, they are usually X-linked because X chromosome carry more gene because they are longer. Okay. And then for males, they only have 1 copy of X from his mother, and then the phenotypes will be caused by a single allele from his mother. And if both parents show the dominant phenotype while the mother is a carrier, and then the daughter will show definitely dominant phenotype, and maybe she is a carrier, but definitely she is alright. While for the son, because the only copy will be from her mother, so it is possible that the son will show a recessive phenotype. Possible, okay? Because the mother will have a correct copy, that's the big A, and another copy is the small a, so it depends on the case, okay? But usually the guys will suffer. So how we can do the inheritance? We can use the pedigree tree. And in the pedigrees related question, this is usually the start without using the genetic diagram, which is the first thing that I told you to learn and told you to draw in this chapter. However, in your exam, you are not and you cannot use it in understanding which chapter is dominant. So here is an example how to describe it verbally. So for curly hair is a dominant trait. Why? Because individual 5 has a straight hair and he must at least have 1 allele of straight hair because it's showing the straight hair, okay? So, and it is inherited from his parents, either 1 or 2. And then both 1 or 2 show a curly hair, which means that they have at least have 1 allele of curly hair. So by combining these 2 information, and 2 must be heterozygous, and with the dominant allele is brain expressed, which means that the phenotype observed in individual 1 and 2, that is the curly hair, is the dominant allele. So you have to recite the format in your exam. So in doing related question, 2 seals are used always true. First of all, look for the mutant or variance. Okay, this is in your first slide I talk about. Okay, an individual show a different phenotype than the parents. Okay, in this case, both parents are green in color, while this one is purple in color. And then you may start working on this individual by understanding that at least 1 allele of purple should present in this individual, and 1 allele of purple should present in these 2 individuals. However, these 2 individuals both show green phenotype, which means that the green allele should be the dominant one. Okay, so how you can do it is to recite the format. Okay, most students can understand the answer by this moment. However, they fail to explain. So the easiest way is to recite the format and then input all the different terms based on your question, and then you can complete your exam. And there are two types of variation. The first type is the continuous variation, known as the heights, and it is controlled by multiple genes, and it is largely affected by the environmental factors, like the food we take and also the temperature or around or so on and so forth. While there are discontinued variation like the ABO blood group, so there are very distinctive phenotype like the A group, B group, AB group, and also the O group. And they are usually controlled by one or only a few genes, and they are limitedly affected by the environmental factors. Okay. And for the cause of variation, like explained previously, majorly will be genetics for the discontinued variation. So how genetic factors may affect it and how the variation can cause. So remember what you have learned in Form 4, that is independent assortment of chromosome at meiosis and also crossover event happen in meiosis. And then in the formation of gametes, there is a random fertilization. And finally, there are mutations may happen in the gamete. However, it only contributes a small amount of the variation comparing with these factors. And for the environmental factors, it includes the temperature, light, and also pH. And a very classic example is temperature, because for the gender of the crocodile, it is dependent by the temperature when they are hatched as an egg. So it's a very classic example how the environmental factor may affect their phenotype. And from chapter 25 to chapter 26, that is from the basic genetics to molecular genetics, you have to understand how DNA works. Okay, so for DNA, it is the short form for deoxyribonucleic acid. For the RNA, is the ribonucleic acid. There is a general structure with a phosphate group and a 5-carbon sugar backbone, and then with a nitrogen-rich base. And here is a comparison table between the DNA and also RNA. DNA is for carrying information, while RNA is for protein synthesis, not photosynthesis, protein synthesis. And DNA is relatively stable, while RNA is relatively unstable. And for the stabilities, it is not included in your curriculum. And RNA has AUGC, which is the main difference, is this T and also this U. And from 25 to 26 chapter, we have this hypothesis, one gene, one protein. That is, for one gene, it may include a structural protein or an enzyme. And then this polypeptide, that is the protein, will be folded properly and then formed as a functional enzyme and carry a specific functions, which means that a gene is determining the body characters based on the protein it encoded. And then the DNA is a large molecule with a lot of nucleic acid, and it contains a lot of genetic information. And it's stable because of its double helix structures, maintained by hydrogen bonds. And then the base pairs are projected inward, which means that they are protected. And then they are pairing specifically to ensure that it is accurate. So here is an extract of the HAEA document. You may notice that codominancy, incomplete dominancy, and also linkage is no longer in your learning and assessment focus. However, some questions in the Asian past paper and also in your book exercise are both sex-linked. And then for your dihybrid crossing, it is no longer required in your learning focus. However, it is still in your book because we think it is a very basic concept. However, in your exam, it may no longer appear. So you may notice that how easy genetics in your lab. And you have to study the pedigree trees and also know the cause of different variations and characters. That is the discontinuous and continuous variation. So this is the end of chapter 25. We understand about the basic of genetics, and in next chapter we will from the basic genetics to the molecular level. So how genetics actually works. Okay, so see you guys next time. Bye bye.